Dear students, we are discussing about the diseases of rice under the course Plant Pathology 5.3. As we know, the rice is very important crop, and we have already discussed a very important disease, that is rice blast. Now, most important disease of rice also caused by a bacteria, that is known as a bacterial blight of rice. Now we will discuss in this lecture about the bacterial blight of rice, its causal organism, different type of symptoms produced by this disease cycle, as well as finally our aim is to manage the disease. This is rice bacterial blight. The disease was first observed in Japan in 1884. In India, bacterial blight first time reported in 1959. A severe outbreak of this disease occurred in Bihar during 1963. Yield losses caused by this disease, generally it is 20 to 30%, and occasionally it will be 50%. In India, millions of hectares are infected every year. Yield losses have been high as 60% in some states and Godavari district of Andhra Pradesh, which are endemic to this disease. The varieties of rice that have Taichung native 1, that is highly susceptible for this disease. Now, causal organism of this disease, as we discussed already, it is bacterial disease caused by a bacteria, that is Xanthomonas origi, PV origi, or you can say Xanthomonas origi, pathovar origi. Bacterial blight, also known as Karaske disease or BLP in short, bacterial blight in English, as well as Sukaro in Gujarati. Bacterial blight appear on leaves of young plant after planting as pale green to grey green water soaked streak. This is the streak caused by the bacteria near the leaf tip and margin. Leaf tip and margin. These lessens colors and become yellowish white with the wavy edge. You can see here, this is yellowish white and wavy edge due to the infection of this pathogen. Eventually, the whole leaf may be affected, become whitish, grayish, and then die. You can observe here, leaf sheath and clumps of the most susceptible cultivar may be attacked. Systemic infection known as Karaske result in desiccation of leaves and death, particularly of young transplanted plant. This is Karaske symptom. You can observe here, whole plant die within a short span of time. In older plant, in older plants, the leaves become yellow and die. If the cut end of the leaf is kept in water, you can see here, it becomes turbid because of bacterial ooze, bacteria coming out from the leaf. And you can also observe bacterial ooze early in the morning when there is a sufficient dew on the infected leaf. Bacteria coming out inside from to this droplet. And you can observe here heavily infected field. Now discuss about the disease cycle of this pathogen or this disease. Here is first, the disease can coming into your field by many sources: disease stubbles, or you can say disease crop residue, disease seeds, and irrigation water. When there is a nearby field infected by this disease, so when irrigation water coming from side by field to your field, it may carry the inoculum of the pathogen. Then how bacteria enter into the plant? Generally, bacteria enter into the plant by natural opening, for example, hydatodes, as well as by a wound created by various factors like insect or brushing by air current. This is, you can see here, this is a bacteria enter through hydatodes, and here it is by enter through wound caused by various factors. Now, the most favorable condition for this disease development is high temperature as well as high humidity and intermittent rain. Then bacteria, after multiplying inside uh, the plant tissues, enter the vein of the leaves. Bacteria that enter the root plug the water conducting tissues, that is vascular bundles, and cause the plant to wilt, and complete plant can die within a short period of time. After the initial leaf lesions appear, bacteria ooze from lesions in presence of free water and spread by rain with high wind velocity in the field. That's why when there is infection in a one corner of field, by short span of time, when there is a rain splash or windy days, rain with wind, it may spread bacteria throughout the field with a short span of time. Then some fact about this disease: why and where it occurs. Why and where it occurs. The disease is most likely to develop in areas that have weed and stubbles of infected plant. It means there is a already bacteria reside in the stubbles or infected parts of the previous crop. It can occur in both tropical and temperate environment, particularly in irrigated and rain-fed lowland area. In general, the disease favor temperature 25 to 34 degrees Celsius and high relative humidity, that is more than 70%. It is commonly observed with strong wind and continuous heavy rain occurs, allowing the disease causing bacteria to spread easily from one plant to other plant and one field to other field through ooze. Bacterial blight can be severe in susceptible rice variety under high nitrogen fertilizers. It means higher dose of nitrogen fertilizer uh, leads to increased susceptibility of the plant for bacterial blight as well as for blast. Then talking about the management of this disease, the first and best option of plant disease management is to use resistant variety. Planting resistant variety has been proven to be the most efficient, most reliable, and cheapest way to control bacterial blight. When you plant the resistant variety, then there is no need to carry out any management practices. Among them, there is few varieties which have tolerant power against this disease, that is IR20, Krishna, Jagannath. These are tolerant variety against this disease. Other factors which we can keep in mind for management of this disease, or you can say other disease control options. These are the cultural options. Use balanced amount of plant nutrient, especially nitrogenous fertilizer, because nitrogen, when you apply excess dose of nitrogen, plant become more succulent and susceptible to disease as well as insect pests. So it is necessary to use balanced amount of nitrogen fertilizer. Second one important, ensure good drainage in the field, conventionally flooded crop and nurseries. It is quite necessary when it need drainage facility should be there. Avoid the heavy disease infection. Then other one factor is keep field clean, do regular weeding, remove weeds, and plow under rice stubble, straw, rice, ratoon, and volunteer seedlings, which can survive as a host of bacteria. Serve as a host of bacteria. These all straw, stubbles, rice, ratoons, these are serve as a host of bacteria. So removal of these material, we can reduce the inoculum level already present in the field. So then avoid clipping of tip of seedling at the time of transplanting. This is very important because when you clip the seedling, it may provide injury or wound where bacteria can enter. Other factors also should also be keep in mind for management of this disease. Avoid flow of irrigation water from infected to healthy field because bacteria can spread through irrigation water. Avoid flooded condition or drying of the field, especially at the time of flowering. Soak the seed in streptocycline 250 ppm followed by hot water treatment at 52 degrees Celsius temperature for 30 minutes. Eradicate seedling infection. Spray streptocycline 250 ppm along with copper oxychloride 0.3%, also quite effective for management of this disease. Now this is recommendation by Navsari Agriculture University. Spray streptocycline plus copper oxychloride in the ratio 1 gram of streptocycline plus 10 gram of copper oxychloride in 20-liter water, or copper hydroxide 50 gram in a 20-liter to manage bacterial blight of rice. Thank you. Thank you much, very much.